Okay, so looks like we've got a nice group for our session. So everybody, welcome to our session at Virtual NERIC 2021 from Neurocircuitry to Neuromodulation. I'm Ben Greenberg. I'm one of the co-chairs, and I am a psychiatrist and originally trained as a neuroscientist. I'm at Brown University, but really Butler Hospital, which is where our COBRE on neuromodulation is based in Rhode Island. And let me introduce our co-chair, Arturo Andrade.
Hello, everybody. I am Arturo Andrade, and I am currently at the Department of Biological Sciences at the University of New Hampshire.
Okay, and do you want to say anything about what you do?
Yeah. So I study mechanisms of alternative splicing, how different splice variants in the brain control transmitter release in specific synapses in the hippocampus, amygdala, and I look into also how splicing is regulated in broad terms.
Okay, so that illustrates a little bit of what we're hoping to show you in this session, which is a very broad session. Now, we heard this morning from the NIGMS director that actually there was a concern that the COBREs in particular were often too narrow, too narrowly focused and the kind of niche in their work. And here we have a number of COBREs represented, other NIGMS funding streams, and as well as people from 4 states in the Northeast region. But what we are going to illustrate in a relatively brief session is a full range of research and building research capacity in ranging from basic science. Our first speaker, Robert Hill, who is going to talk about really developing the wiring of the brain, myelin. And then we're going to go to behavior in an animal model. Amanda Crocker at Middlebury is going to be talking about how to measure changes in Drosophila in fruit flies in response to painful stimuli. And again, the understanding is there how circuits function in response to very behaviorally meaningful contingencies. Then Oriel FeldmanHall is going to talk about navigating our uncertain social worlds, talking about really the cognitive and social cognition and how one uses information in part, I think, to make decisions, to navigate those uncertain social worlds. She is in the COBRE on central nervous system function at Brown University. And then finally, we're going to, sort of the culmination of this work is going to be by Mascha van Wout Frank, who is in our COBRE, the one I head, and is co-led by Linda Carpenter at Butler Hospital, which, by the way, blocked this website apparently, but now we should be able to get in, on doing neuromodulation studies, clinical studies in humans. So I think, and after that, we're going to have a pretty brief discussion, really more a Q&A period, but Dr. Andrade and I may have some remarks based on what we hear today. And so again, we're hoping to show the full range from basic to translational. A lot of things are meant by translational research to clinical research in this session. So I think we'll get started, and I hope, Arturo, you can introduce Dr. Hill.
Yeah, of course. Hi, everybody. It is with great pleasure that I'm introducing Dr. Robert Hill. He will share his experience with these interactions between neurons and glia. I'm very much looking forward to hear what he has to teach us about these important interactions. And the stage is yours.
Okay, great. I want to thank Ben and Arturo for inviting me to give this talk. I'm very excited to tell this audience about some of our work. And as Ben was mentioning, we'll be going in this session, you'll be going all the way from cells to clinical human trials or human work. So I'll be starting with the cells. And I would hope to remind you as you're learning about the clinical work, think back about what the cells might be doing in that context. And that's that. Keep thinking about that when I'm talking about stuff throughout my presentation. So as Ben mentioned, I'll be talking about myelination. Here's an example of an oligodendrocyte, a myelinating cell in the central nervous system. And you can hopefully appreciate the beauty of these cells. And so I first, when I was a graduate student, got exposed to these cells, and I have to study how these cells develop, how they make these elaborate structures, and then how they may change over time. So to give you a real brief introduction about myelination, so we're all kind of on the same page and kind of give you an idea of why you should care about this, we can think about a neuron in your brain. It could be a neuron in your cortex, in the cerebral cortex, or some neuron that's projecting down through the spinal cord. And you can think about these cells projecting to many different areas. And what I've diagrammed here is just a simple axon that's coming out of that neuron, and it's projecting to two different targets. And you could imagine in different types of neural circuitry, you would want action potential or a signal that's generated in this neuron to arrive at these two targets at the same time, even though they may be projecting different distances. And so one strategy that the nervous system has developed to achieve this is myelination. So this is just obviously very simplified, but you could have this branch of this axon be myelinated. And what the result of that would be is that the propagation of a single signal that's initiated in this cell will arrive at this target and this target at the same time, even though this one may be projecting a much longer distance. So it's really, you can think about the insulation of your axons in your brain or your wires in your brain. And I'm very interested in this for many reasons, but one of them is that they're one of the only populations in the central nervous system that maintains a progenitor pool throughout life. And I'll be referring to these cells as NG2 glia. They're also called OPCs, oligodendrocyte precursor cells. And these NG2 glia throughout life, very robustly during development, but also throughout life, are continuously generating these mature oligodendrocytes that make this myelin sheath. And there's a lot of interesting questions about the signals that are inducing the generation of one of these new oligodendrocytes. And then we also have some questions about how these oligodendrocytes decide to myelinate some axons and not others. It's not that all of your axons are myelinated in your brain, it's actually a subpopulation. And then there's also many questions about once you establish one of these mature myelinating oligodendrocytes, is there any plasticity of these cells, structural plasticity, which then could potentially modulate that signal that's propagating down that axon? And then finally, also, when you generate a new cell, especially in the adult brain in a somewhat already established neural circuit, how do they interact with already established cells, with already established axons? How do they decide where to myelinate which axons and at what time? And so there's a lot of kind of baseline plasticity mechanisms that I'm very interested in understanding. And so there's obviously a lot of molecular signals that induce each one of these steps, how this proceeds developmentally. And then you can imagine a lot of different diseases such as multiple sclerosis, Alzheimer's disease, and some of these other psychiatric disorders that I have listed here are implicated and have myelin disruption. And so it's very important for understanding some of these basic cellular plasticity mechanisms in order for us to understand what goes wrong in these different diseases. And so all I've showed you so far is just what these cells look like as diagrams, but we use mouse as a model for understanding some of these basic cellular mechanisms. And here's just on the left here, we have a cross section through the mouse brain, and we have the CMPase that's labeled showing you the myelination in the brain in green. And so you can see there's quite a bit of myelin in the brain. And also I've mentioned that oligodendrocytes are the cells that make these myelin sheaths. And here's an example of a transgenic mouse that we have that labels single oligodendrocytes. And as this would actually be the cell soma, or the cell body of this oligodendrocyte. And you can see that one cell actually can make up to like 60 different myelin sheaths. And many of these will be on different axons. So it's a very elaborate cell structure. And as I've brought up, I'm very interested in how this gets set up and how it changes over time. And so how do we do this? Well, we use live imaging. Mainly, most of the stuff I'll be talking about would be live in vivo imaging in the mouse cortex. And so we do this fairly simple surgical preparation where we perform a craniotomy, which that means that we just remove a small piece of the skull and replace it with a cover glass. And then the mouse can go back into the colony, and we can use confocal or 2-photon fluorescence microscopy to follow individual cells over minutes to months, and kind of to understand how these cells change, you know, all these basic properties that I've introduced to you, and then also how they may just be disrupted in different models of disease. And so we use fluorescence techniques, confocal and 2-photon, and that's what you're seeing on this slide. We have on the left here, we have just some different labels, fluorophores that are targeted to the mature myelinating oligodendrocytes, and we're just taking individual slices down into to the brain of the animal. And again, this is the live animal. And this is just a single time point. You can see this is the depth going down into the cerebral cortex. But the real power of this is going back to these same cells over time. So that's what you're seeing on the right here. Some of those NG2 glia, those oligodendrocyte precursor cells. And you can see that these are highly proliferative cells in the brain, and you can see a cell divide and then divide again. And this is over 26 days. So we're going back at each one of these time points, finding this cell and looking at its dynamics. We've also, and I'll just briefly touch on this, but we've also developed a label-free technique for visualizing this myelin structure, which we've called SCORE. That stands for spectral confocal reflectance microscopy. And so we're still using a confocal microscope that many of you might use if you're doing cellular biology. But instead of having it in fluorescence mode, where you're exciting a fluorophore and detecting the emission, instead you just have it in reflection mode, which is exactly what it sounds like. Let's say you send down your 48 nanometer laser and you collect 48 nanometers. When you do that in the brain of a mouse, you actually get these extremely highly reflective fibers. And these fibers we found are actually exclusive to the myelination in the brain. And we use multiple different wavelengths of laser because actually you see that different fibers or different even parts of that fiber will predominantly reflect different wavelengths. And I won't be showing many colors like this for the future slides because it gets overwhelming when you combine that with the fluorescence. But just so you know, when I'm showing SCORE, it's actually a combination of many different wavelengths combined into one channel. And so what this allows you to do is get images like this, and again, in the live animal, and you can do this with label-free techniques. You can take a wild type, animal, you can take human tissue, you can take a zebrafish, you can do this in a lot of different animals that have this myelination. And you can see each one of these fibers now, we're looking now onto the surface of the brain. Each one of these fibers is a myelin sheath. And then we just labeled the blood vessels here for kind of context. But we can use this again to visualize myelination in a label-free fashion. And just to give you a little bit of idea of the specificity of this, here we have a single one of those axons, one of those wires that I've introduced in the introductory slide. And you can see there's a bifurcation of this axon. We've labeled this axon with a fluorescent protein. And then at bifurcations of axons, we know that there's a break in the myelin sheath. There's often nodes of Rambier here. We can visualize that by putting a dye that actually labels the myelin. But if we go to our reflection technique, our SCORE technique, you can see it perfectly. There's no signal that's arising from this bifurcation. But as soon as the myelination starts, we get this label-free reflection signal. And you can see it also here on the bottom of the slide, where we have these actual proteins that we've stained and labeled in tissue, that we know that this is a node of Rambier, this is a node of Rambier, and you get this nice reflection signal between them, indicating this is the myelin sheath, and then you have a break, and then we have the axon labeled. So it's very specific. And so if we have a label-free specific technique like this, and if you're interested in all these questions about how plastic these structures are, how long does this generation of new cells occur in the adult brain, you may take all your animals in your colony. If you have a mouse colony of different ages of animals, you're like, well, we have this label-free technique. Let's take a look and see if any of these structures change over time. And so that's what you're seeing. These are all different ages these are different animals here. It's going all the way from 30 days old all the way up to 950 days old. And so you can see this is all looking in the same region of the brain. So presumably if there's nothing changing, there would be no changes in this density. But we, as you can appreciate by these images, we see pretty significant changes in the density of these reflective fibers, going almost all the way up to 2 years old in the mouse brain, which is quite old in a mouse. And you can see this, that's quantified down here. We don't need to get into the details of that. But we can also take, the one question you may have is this: well, are these mature ligadendrocytes making new myelin sheaths? Or when you see this change in density, is it actually that there's whole new cells that are there coming from, probably from those NG2 glia progenitor cells? And so we can label the mature ligadendrocytes and then just quantify the density of these cells. And you can see that actually the density of these mature ligadendrocytes changes pretty much corresponding to the density of these myelin sheaths, suggesting that as this myelination density increases, as you get more and more myelin, it's actually due to mainly to the generation of whole new cells in the adult brain. And so those are different animals. We're taking cohorts of animals at each one of those stages. But as I mentioned, the power of this is going back to the same animal, the same cell over time. And so that's what we're doing here. We can directly get at that question. When you have a new myelin sheath pop up, is it that there's a new oligodendrocyte that's there? And so here's just an example of this, a same animal that we've imaged at 50 days old. You go back 60 days later, you can see here's some of the same fibers, but there's tons of new fibers there. And if we use one of our transgenic animals that labels the mature oligodendrocytes, you see that actually every time that there's new fibers, there's a whole new oligodendrocyte that's popped up. So that kind of we see a brief overview that there is a kind of this lifelong addition of new oligodendrocytes in the cortex, at least the superficial layers of the cortex of the mouse. But what about the individual oligodendrocytes? Are they able to change their structure over time? And so we can use some kind of tricks of mouse genetics or transgenics to label individual cells. And so that's what you're seeing here. There's a single mature oligodendrocyte. We've labeled it 60 days old, and you can go back to 60 days later, and that's still that cell is there. We can zoom in on one of these myelin sheaths and look to see, okay, are they able to change their length over time? And you may say, why do we care about that? Well, modeling studies show that the length of the myelin sheath pretty significantly alters how quickly the signal propagates down that wire, down that axon. So pretty small changes, micron changes here, can have, at least in terms of modeling, we don't have a total functional readout of this yet, at least in cohort with, or at the same time as this type of imaging studies. But it would suggest that there's pretty significant changes in the propagation of the signal down that axon. And so I'm talking about it because we did see some significant changes in length. Many of these myelin sheaths are very stable, almost 80% of them at this age of the animal. But we did see extension, we did see retraction. And even at the individual cell level, there are individual myelin sheaths, some are getting longer and some are getting shorter, suggesting that it's a direct interaction between the axon and that sheath. And it's not that the whole oligodendrocyte is getting bigger or getting smaller. So we do see that there actually is the capability of plasticity of single myelin sheaths. But now what if we take the perspective of the actual individual axon? And so there's a very nice study a few years ago now that looked at myelin patterning down single pyramidal cell axons. And so they use 3D electron microscopy reconstruction. And you can see along this single axon here, this part they found was myelinated, then there'd be a big break, and then it could be myelinated again. There's other axons that are, from other cells that are not myelinated at all. And then those ones that they don't get myelinated until a ways down from the cell soma of that neuron. And so this is kind of a new way of thinking about, when you learn about myelination in textbooks, you're like, okay, there's a myelinated cell and then there's non-myelinated cells. And you think of it as fully myelinated, but this is showing that that's not the case. And what we thought, okay, so this was all at one age in P60 in the animal. What's happening to these bare regions over time as we're seeing the addition of new cells? And so is it possible that when you generate a new oligodendrocyte, is it myelinating some of these non-myelinated regions and filling in some of these gaps throughout life? And that could potentially modulate the action potential propagation. So we can use some of our techniques. We can label the axons. We can use our SCORE technique, look at distribution of myelination down these axons, and we can see the same thing. There's portions that are myelinated, portions that are not myelinated. Get some kind of nice reconstructions of this in our cranial window preparation. And then you can go back to those same axons and those same regions. We don't need to get bogged down by the details here, but it's kind of the evidence of this. You can see if we look over on the right side here in this panel E, there's this one axon, had a couple myelin sheaths on it. Then we go back a few days later, a few months later, and we can see, yeah, there's a new myelin sheath here. There's also a new myelin sheath here, and you can see that from the raw data here. And so this is suggesting that, yeah, actually over time, there is, when you generate a new oligodendrocyte, it's likely myelinating axons that were partially myelinated, but also some that maybe were never myelinated to begin with. And I should also mention that P60 is often used as an adult mouse. So in terms of myelination, that's kind of just, at least in the cortex, kind of just getting started, at least upper layers of the cortex. And so again, keep this in mind when you're thinking about how different manipulations in animal models or in humans, when learning and other things, whether or not myelination could be involved in that. And so we can do a little bit more quantification of this and look at distribution of myelination down axons at many different ages, kind of similar to that those ages that we were looking at in terms of the myelin density throughout life of the animal. We can see that over time, there's more and more myelin coverage along these single axons. There are still some axons in the really old animal that only have a couple myelin sheaths on them. So it's not that everything gets filled in, but again, showing that there's probably some specificity to this that we don't really understand yet. But over time, it seems like there's more and more filling in of these gaps. And what the reason for that is, we don't know. Do you always need to keep increasing the propagation of your signal, or does this have some other function? And we'll touch on that a little bit at the end. So what we can kind of conclude from this part, if we go back to our diagram that I introduced to you at the beginning, we have this kind of lifelong generation of these new oligodendrocytes. We do see that there is some capability of internode plasticity or myelin sheath plasticity, which is kind of the first time that that's ever been realized or visualized, and the implications of that and how widespread that it might be in the human brain are open questions. And then there's some lifelong changes in this patterning of this myelination down single axons. Well, a lot of our motivation for doing this, as I mentioned, is understanding different diseases. So can we use this type of model then to understand what happens in demyelinating diseases or in aging and other situations where you have loss of these myelinating oligodendrocytes? And is it possible that, it's fairly well known that you can repair some of this myelination or replace these cells through these progenitor cells, but can we model this and use some of our imaging techniques to understand some of the cellular dynamics of this? And so to do this, as you would imagine, there's many different mouse models that mimic or can induce demyelination, but many of them you can't choose which cells you want to kill, and it's not localized to, say, our cranial window or our in vivo imaging region. And so we kind of tried to come up with a way to induce the death of individual cells. And so I'll go on a little bit of a tangent here talking about our method for inducing cell death. And we've called it 2-fatal, which stands for 2-photon apoptotic targeted ablation. And it's actually a pretty simple technique. While we're doing our cranial window procedure, or after we've done that, we apply a nuclear dye, this Hooks dye that many people are probably using in cell culture studies. It very nicely labels the nuclei of all cells. And so you can see here's an example of some neurons that are labeled in a transgenic animal that has subpopulation of neurons labeled. And then you can see all the nuclei labeled within our little imaging region. And you can see nicely here's that single nucleus of that neuron. And what we do is we take a 2-photon laser, we choose, okay, let's say we want to kill this cell, and we want to kill it in a somewhat physiological manner. We don't want to just blast it and burst open and cause massive inflammation and everything. We don't want to actually induce apoptosis or controlled cell death of that cell. So we choose, let's say we want to kill this cell. We actually focus the laser on that cell. We do a subtle bleaching of this nuclear dye, this dye that's intercalated into the DNA. And we found that over time, the idea there is to cause some subtle DNA damage, and that would start the process of apoptosis in the cell. And we found that it works quite robustly in many different cell types. So here's an example of this cell. And then you can see we've gone back to it over time, and it condenses, the cell nucleus condenses, and then by 18 hours, it's gone. And here's just an example of a cell that we targeted. You can see all the other tissue around it. There's actually, it's this one cell is gone, but the other processes of other cells and the neighboring tissue doesn't really seem to be affected much by this. And you can use this to study all kinds of different things. Here we're labeling the mitochondria. You can see mitochondrial fission during the cell death process. We can target other cells. Here's cells that are associated with a vasculature in the brain, where you can induce their death and look at how that might change vascular structure and other things. So it's kind of, we've developed this for many different cell types, but really my motivation for developing it was to understand demyelination. And so, well, as an aside, also we could use it for studying cell-cell interaction. So here's an example of a neuron that we've targeted. We've induced the death of it. We have microglia, the macrophages in the brain, that actually jump on, and when the cell is dying, they start clearing it up quite rapidly. And this is, again, this is all in the live mouse brain. It's actually an awake animal that this is happening, that we're imaging this. And we have a whole paper kind of describing this process. So it's a pretty powerful method for inducing the death of individual cells. And so what about oligodendrocytes? Well, we had a lot of struggles with this because there's some interesting properties of oligodendrocytes that we don't totally understand, but we do this same 3-second, 5-second photobleaching of the nuclear dye in an oligodendrocyte, and 45 days later, the cell finally dies. So in neurons, you notice it was, it's 24 hours, the cell would completely be gone, which was really nice for our experiments. Oligodendrocytes, I kept thinking they weren't going to die just because they kept hanging out. But it ended up being that I didn't wait long enough until my graduate student was, was like, oh, we need to, something looks a little bit strange about these cells. We should keep looking at these animals, and hopefully our cranial window preparation lasts that long. And so that's what you're seeing here. Is, this video is looping, and you can see these are days after the 2-photon photo bleaching. And here's the oligodendrocyte. And you can see it has some morphological changes. It starts to condense. And then finally, I think it's on 40, 41 days here, the cell disappears. And interestingly, all the myelin sheaths that are attached to that oligodendrocyte, you can see them degenerate. And then a lot of them, you can see them get regenerated, actually even sometimes before the cell totally disappears. So there's tons of dynamics going on here that allow us to study this process in a lot of detail, again, in the intact brain. And so just to kind of give you a quick idea of how robust this method is for inducing the death of cells, here's a % survival of cells that we targeted. You can see they may last about 45 days, about 50% of them will die by 45 days. And then 100% of the cells that we target with this photo bleaching end up dying. And we can compare this to another model where you feed animals with this toxin called cuprosome that actually causes some selective killing of oligodendrocytes. And you can see, at least for this one experiment, about 50% of the cells died in our window due to this cuprosome. So, and you can actually see there's some similarities in terms of the cell condensation and the loss of the cells. And I won't get too much more into that, but it seems to be replicating some other models that induce death of oligodendrocytes. But what's so nice about this is 100% of the cells die, and we get to decide, we want to kill one cell or do we want to kill 30 cells within our cranial window. And to kind of hopefully convince you that this is not a completely wacky thing that we're doing. I mean, it is a little bit, it's not physiological. You're not having nuclear dye in your brain that's getting photo bleached to cause the death of your oligodendrocytes. But the outcome of that seems to be very similar to what we see in the aging mouse brain. And so here's just some morphology of single myelin sheaths that we've targeted that are attached to our cells that we've targeted with 2-fatal, where we can see this kind of blebbing, some regions where there's none of that score signal. And if we look in the aging brain, you can actually see as these cells are dying due to age-related processes, you can see some very similar structures. And we have more characterization of this. So we really think what we're modeling here is not like multiple sclerosis. We're modeling some type of oxidative stress, DNA damage mediated cell death that probably is what's happening in the aging brain. And so we can quantify a lot of this, get a lot of information about the degenerative process. And you can see that all the sheaths that are attached to that cell eventually degenerate. They don't all degenerate at the same time, which is interesting. But the vast majority of them degenerate before the cell soma degenerates, which suggests that there may be some mechanism that's helping support those over those 40 days, helping support those sheaths over those 40 days. And then you can see here's an example of a single sheath degenerating over time, kind of going through some of those stages that I mentioned on the last slide that looks very similar to what you might see in age-related degeneration. And so we're obviously very interested in, we want to know how this degeneration proceeds, but maybe more interestingly, we want to know if it can be repaired, and what can those distribution or patterns of myelination down single axons, can they be restored, or how important might that restoration be for the function of the cell? And so we can use some different dual labeled transgenic animals. I again won't get into the details here, but here's an example of one of those progenitor cells. You can see it actually differentiate or turn into an oligodendrocyte, start making new myelin sheaths. And so then we can use this mouse line then to understand how these progenitor cells go in and myelinate one of those sheaths that degenerates, kind of getting a little bit more information from that video that I showed of the oligodendrocyte degenerating. So here's an example of one of those sheaths. So this is before we've done that two fatal photobleaching. You can see the sheath is degenerating, and then it's gone. And then now we have this other labeled cell, this NG2 glia, this progenitor cell, comes in, reaches out a process, starts making a new myelin sheath that now is dual labeled so we can know that those dual labeled ones are the new remyelination that's occurred. And you can see, even if you look, there's this little break, it actually gets reestablished. And that node of Ranvier is actually probably in a very similar location to where it was originally that happened during developmental myelination. So this one example suggests that you can actually potentially reestablish some of those patterns. And to just kind of give you an idea of the different outcomes that we might see, there can be some sheaths that, that were not targeted, not attached to a targeted oligodendrocyte, and we can see that they're stable. It's not dual labeled. Some sheaths that are lost that were attached to the targeted oligodendrocyte, but they never get remyelinated, at least during our imaging time frame. And then there are some that get remyelinated, they get double labeled. And so we can use that, this dual label here, in addition with our imaging techniques, to get a little bit more information about that remyelination and what's more or less likely to be remyelinated. So we can just simply look at individual axons. And as I mentioned, there'll be some that are partially myelinated. Maybe there's a single sheath here that is not flanked by two other sheaths. There's some that maybe has one sheath next to it and the other side is not has bare axon. And then some that are completely myelinated within our imaging window. And what we've observed is actually if you do two fatal on all of these different situations, you can see the percent that are likely to get remyelinated are much higher if it's a partially myelinated or completely myelinated axon. So again, you're kind of reestablishing some of those patterns down those axons. That seems something intrinsic likely to this axon that says, sending a signal, say, I need to be myelinated. And this happens much faster actually than in the situation where it's partially myelinated or completely myelinated. And so I'm actually going to skip this part just based on time. And then we can also then do this in aging animals. So I keep bringing up that this is probably replicating aging, but why do we care about that? Well, one reason is that there's evidence that in aging humans and in late stages of multiple sclerosis and late stages of Alzheimer's, there's actually death of these oligodendrocytes, and then remyelination does not occur. So what if we do this two fatal in these aged animals? Do we actually see the same likelihood, the same remyelination occur? And it would give us a very nice model for understanding at the single cell level what the mechanisms are that are inhibiting that remyelination. And so we can do this two fatal. It works just the same in the aged animals as the young animals. You get the same death progression of these single cells. But the big difference we saw is that if you targeted a oligodendrocyte in a young animal, again, you can see massive remyelination by those progenitor cells. Whole new oligodendrocytes are generated here. You can see in this window. Whereas in an aged animal, we've targeted this cell, it dies in the same temporal process, but we see almost no remyelination. Again, saying that this is replicating what's likely happening in late stages of disease and in aging, and we have a nice model to visualize that now. And so what have I told you kind of through a slew of many different images? Well, at least in this two fatal model, this oligodendrocyte death is slow and unique. And so we have a whole set of experiments kind of understanding why does a neuron die in 24 hours where the oligodendrocyte takes a month and a half to die from the same insult. And that can be quite important for in many different contexts. This response by these NG2 glia, these oligodendrocyte progenitor cells, is local and heterogeneous. So I didn't get into much of that, but we have it in our preprint here. And then these patterns of remyelination, the patterns of myelination down these single axons depend on the initial pattern. And there's this form of rapid remyelination. That's the part I skipped over that you should definitely check out in the preprint. And then this remyelination is defective in aging, even at the single cell level. So if we think about what this might mean and why do we care about this? Well, there's quite a bit of evidence in the literature that things like sensory experience, different environments, all of these kinds of different situations can modify this process. We didn't know really at the cellular level exactly, or are they changing the lengths of the sheaths? We didn't have the tools yet to visualize that, but there's quite a bit of evidence that neuronal activity and sensory experience are important in setting up this structure and potentially even in the adult as part of a learning and memory type mechanisms. So we now can take models that manipulate those things and use our imaging techniques, these are the things we're doing now in the lab, to kind of understand how sensory experience is involved in setting some of these patterns. Maybe it's some of these addition of new myelin sheaths late in life or early in life are important for closing critical periods of plasticity of the axon, all kinds of other potential implications besides just action potential propagation. And then obviously implications for remyelination and whether we can boost these progenitor cells to reestablish some of these patterns late in aging. And so with that, I'll end and happy to take questions. I should acknowledge we're part of a COBRE grant by Dean Madden as the PI. It's called BioMT. So I had a pilot grant from them, and that was very useful when I was first starting my lab a couple of years ago, and also for other funding mechanisms.
All right, this is terrific. I mean, we don't have a lot of time before the next talk, but for me, your talk raised lots of questions. In particular, I'll just ask one, and then please other people add them in the chat or others. We are particularly interested in a number of different kinds of functional brain networks. So, the default mode, salience, executive control network, they have homologs in the mouse. Do you have any reason to believe that the plasticity of the myelin-related processes that you study might differ across functional brain networks? So, do you want to take a stab at that one?
Yeah, I'm sure they are. And the interesting thing is I'm always going to databases where they show the projections of axons, right? And trying to understand, okay, this one's projecting here, but we never know if they're myelinated. I mean, sometimes you can generally say, oh, this axon is likely to be myelinated, but I think that's a huge area that we should study, like, oh, these cells are active together, these different regions are active together, setting up some of these default mode or whatever. That those could be myelinated in different patterns than other ones. And more, I think, even adding to that is what if you demyelinate some of those axons, how much that could actually mess up the synchrony of some of these cells firing together and their arrival of the target, their signals arriving at their targets. So I think that's that's one what we should be getting into, is trying to connect the wire, the insulation on the wire with the wiring diagram, and what the patterns of this myelination might be. As I mentioned, that paper from 2014, that science paper, that was kind of one of the first where people widely realized that it's not fully myelinated. These axons are not fully myelinated, like the textbooks say. So I think that's definitely the future, to understand which populations of axons are myelinated in what patterns.
Okay, we have one in the chat. Arturo, do you want to read this?
Yes. I was about to do that. So we have this question saying, is myelin protective against too fatal, and do oligodendrocytes have increased DNA repair capacity, or are different apoptotic pathways active in these cells? It's a very loaded question.
Yeah. Well, that's the graduate student that put this, my first graduate student that's the first author on this preprint. That's his real thesis project, right? Is understanding this. So yeah, it's all those things are possibly involved in this. I don't know about, well, the thing is, it's not, I should mention that there's no protective thing here. It's just different temporal dynamics, right? 100% of the cells still die, but maybe it can help it survive. Maybe there's something about myelination, just the lipid content or other things that can help it last longer. You would think that actually might be the opposite of that. So some of these things are a little counterintuitive that we would expect. But yeah, in the DNA repair capacity, that's totally possible. I think the one thing we're thinking is actually it's more like different apoptotic or other controlled necrosis or something like that that's happening within these cells. But we can't find actually anything in the literature that shows a cell death process that's a month and a half. So we're pretty excited about it, but it opens up all these different things that we need to cover. But hopefully, it's a good method for inducing that, and hopefully, we're not just investigating what 2-fatal does. It's what's happening in aging and other contexts where you have cell death of oligodendrocytes.
There's a question from our next speaker, so I don't feel bad about delaying her talk. Amanda Crocker says, do you see behavioral changes? I had this question too. Due to demyelination during your targeted death assay, is there an amount, like a threshold, you need to demyelinate before you would get any?
Yeah, I think with our 2-fatal method, we're never going to see behavioral effects just because we're killing a single. The most we've done is maybe 50 cells, which is nothing in terms of the animal. So in other models like Cuprazone and those models where you have more massive widespread demyelination, you definitely see behavioral effects. And so I think using our system in combination with those other demyelination models is probably the best strategy. This is really just nice for the cell-cell interactions and the cellular dynamics, but it would be great. I really want a functional readout actually of what happens. And so maybe, the physiology of the axon is kind of another way we're going to go about this, not behavior of the animal, but what happens to that axon if you chronically demyelinate it, and what are the signals coming from that axon to induce this rapid remyelination. So if I can get some functional readout, or if you have some ideas for some functional readout, that would be awesome. But we don't have behavioral yet.
Well, I think we do want to move on to Dr. Crocker herself and her talk. This was great. And, let me just make the comment that if there are other interactions that can happen as a result of these interesting issues being raised or by any participant, then we more than encourage and hope that people will get in touch. So Arturo, please take it away.
Yeah. So it's also a great pleasure to introduce Dr. Amanda Crocker. I apologize if I bastardized your name. I just don't know. So she studies one of my favorite animal models, actually, Drosophila. I love fruit flies. And she focuses on understanding the molecular underpinnings of complex behavior. The talk is about pain today. Boy, that is a complex behavior. So I look forward to your talk, Dr. Crocker.
Thank you. All right, let me get this up and going. Okay, are we up and good? Okay. So I would like to thank the organizers for allowing me to come and tell you about the work that we've been doing in my lab to understand the behavior and the behavioral responses due to painful stimuli, in hopes that we will gain insight into some of the underlying circuits. So today I'm going to tell you a little bit about why we're interested in pain, specifically pain in the fruit fly, and try to address some of this question of, does the adult fly feel pain? I think this is a really important question when we talk about pain, and especially in model organisms, when we have animals that don't actually tell us that they're in pain. So we need to use a number of other measures to determine whether or not they're actually feeling pain. So how do we do this? What are our behavioral measures? How do we quantify this in the fly? Most of the work that I'm going to tell you about is dealing with specifically electric shock. And interestingly, electric shock is really not well characterized in any model organism. or even in humans for understanding exactly why it's painful, what pathways are used to actually make it painful, why it's perceived as painful. And then I'm going to end with some of our beginnings to basically map the circuit in both larvae and the adult fly that are important for this type of pain perception, so electric shock, and the behavioral responses to electric shock. So why pain? We know that pain has a very large both fiscal cost for the United States, as well as also a major health issue for many people in the United States. So we know that 1 in 5 US adults experience chronic pain. If you're suffering from chronic pain, that makes you significantly more likely to develop anxiety and depression. The total cost of chronic pain is more than that of cancer, diabetes, and heart disease. And approximately 58% of chronic pain sufferers are treated with prescription medication. And so these are all always really big reasons for trying to understand more about pain pathways, and in particular, some of the genes that underlie pain perception. So one of the things that I'm particularly intrigued by is that pain perception is very variable. So we know that there are many things that modify one's perception of pain. And this includes age, exercise, race, ethnicity, sex, socioeconomic status, and emotional response. And interesting, some of these things also affect model organisms. And we get differences in variability within the same model organism across different sexes, if we stress them out, things like that. I also want to point out that our own perception of pain biases our perception of pain in others. And this has been sort of well documented. It's well documented in the clinic, but it also affects those of us who study model organisms, because we have this perception that something should be painful, and we think that the model organism should also be feeling pain when they experience that stimulus. And so one of the things that we have been focused on is using some machine learning and some of this sort of deep learning algorithms to basically do really fine mapping of behavior to try to basically remove ourselves as the researcher in quantifying, is that a painful response? And you'll see when I talk a little bit more about some of the responses that we would normally behaviorally characterize as a painful response don't necessarily mean that it's a painful response. So most painful responses elicit a startle response, but things that are not painful also elicit a startle response. And this is true for also if you're studying rodents and they're lifting their paws, that doesn't necessarily mean that they're feeling pain. It can be a startle response, even if you think that the animal should be feeling pain. So what is a noxious stimulus to the fly? How do we know whether it's painful? Our fly is not telling us this. So we are using behavior as the major sort of readout of whether or not that stimulus is painful. And the one that we use most extensively is behavioral avoidance, right? So does the fly choose not to be in that area of an arena where that painful stimulus exists? Okay, do they move away from it? Do they elicit an escape response? Okay, so when this painful stimuli comes on, okay, do they then try to fly or remove themselves from it? Do we see learned avoidance? This is fear learning. And we have many decades of fear learning in the fly as well as in the rodent using electric shock. Okay, so where we can pair a noxious stimuli with a more neutral stimuli and ask whether or not the fly avoids that, or the animal in general. More recently, there's been a lot of work looking at stress behavior in the fly. So whether or not the fly shows what's known as thigmotaxis, so similar to an open field test in rodents. So where they're put into an arena and they move towards the walls to sort of either try to protect themselves. Flies do a very similar thing. They like to follow the walls, particularly in times of stress. Do we see activation of sensory neurons that we already know carry noxious stimuli? And so, you know, we've also had many decades of looking at pain research in the fly larvae, characterizing a number of genes that are really important for noxious heat. And so do we see some overlap between those heat stimuli and the shock? So before I go into sort of great detail about this, I'm going to give you a little bit of history on sort of how we came to this. And it will make sense because we ended up with a mutant that had kind of a strange phenotype. And that's what kind of led us down to this road of trying to understand what pain actually looks like in the adult fly behaviorally. Okay. So this started when we were screening for memory mutants. And so this is aversive learning and memory mutants. Okay. And so we had taken this neuron, it's known as the doll neuron, and we'd harvested out of the fly brain. There's 2 of these doll neurons in the fly brain. They're found bilaterally. They're large neurons. They're known to be peptidergic. They've also been implicated in learning and memory. And what we did is we harvested them and we did an RNA-seq experiment where we asked what are the genes that are differentially expressed in this neuron following the formation of an aversive memory. And so we had a number of genes that were upregulated and a number that were downregulated. And one of the ones that we focused on was this NINA-A gene that's shown here that's downregulated. And when we looked at the mutants for this, what we found is that they had a slight deficit in their immediate 10-minute learning, but it wasn't really extreme. The numbers probably would clean this up. But at 24 hours, they really showed no evidence of any sort of persistent long-term memory. And so we were really kind of curious, okay, so they aren't able to form this long-term memory, why? And so when doing these type of learning assays in flies, one of the things that you always need to check is you need to make sure that they can detect the odors that are being paired with the electric shock or whatever stimuli you're giving with the electric shock. And you need to be able to also test whether or not they actually avoid the electric shock. And so this is our shock avoidance assay. This is a watch glass. We load about 16 flies on here. We have a shock side and a no shock side. We'll turn on the shock and we'll let them go for 10 minutes. And then we kind of track where they go. Okay. And now what we're doing to further this is we're now taking individual flies and we zoom way in on them using high-speed video recordings to actually look at the really fine behavior of individual flies. Okay. But in this group assay, we then break it up and we break it up into these zones where we have a no shock inner zone, a shocked inner zone, an outer shock zone, and a no shock outer zone. And these outer zones are to quantify that stress behavior that I mentioned before, okay, and that wall following behavior. So what we found was when we looked at these NINA-A mutants is that they showed very little avoidance of that shock area. In fact, they pretty much didn't seem to care. They kept entering it and exiting it, okay? Whereas their wild type controls shown here, these Cantonese flies, were pretty good about figuring out that this is the no shock zone and spending most of their time there. So what is NINA-A? NINA-A, it's a cyclophilin, okay? We know that it's involved in protein folding and trafficking. It's been most well characterized in the eye because we know that it's really critical for trafficking rhodopsin 1 in the eye of the fly. We also know that it's part of the apoptotic signaling pathway in response to ER stress. We know that it localizes to both the cytoplasm and the ER, and it has an ortholog in humans. But both the ortholog in humans and NINA-A itself are really not well characterized. And for the purposes of this talk, I'm not going to tell you that much more about actually the characterization of NINA-A and what pathways it might be involved in. But I do have that data. If anyone's interested, they can ask me about it afterwards. What I'm going to spend the rest of my talk sort of talking about is why are they failing to avoid the electric shock, and how do we know that? Okay, so why do they fail to avoid it? There could be a number of reasons why, right? It could be an input issue, right? So within the sensory, they just aren't detecting it, right? So it's just failing to get to the brain or to higher brain regions where it could be integrated, okay? It could be an integration issue, right? So is the animal actually feeling that electric shock, but then failing to integrate it in the proper way? Is it purely like an output? They just can't figure out exactly where they need to go. They know they need to go somewhere, but they just can't figure it out. So we started initially to ask, okay, do they have detection deficits? And the first thing we did is to ask whether or not they jump in response to electric shock. So if we turn on the electric shock, do we see that the animals produce this sort of escape response, this jump response? And we see uniformly the NINA-A mutants all jump along with their wild type controls. So Canton-S here and white 1118 are their sort of background controls. And what you can see as well is that the rhodopsin-1 mutant also elicits this jump response. What I didn't show you is actually that rhodopsin-1 animals are perfectly fine at avoiding electric shock. So even though they're blind, they can avoid electric shock just fine. So we know that it's not through rhodopsin-1. So then we said, well, is it not just an electric shock deficit? Do they also have issues with temperature? And so the fly larvae has been used for many years to study noxious heat, and they have a series of very well characterized behaviors to determine their ability to detect heat. And so we took the larvae and asked, when do they elicit full body rolls, which is part of this sort of escape response at different temperature onsets. And what we find is that our NINA-A mutants really don't look that much different from their background controls. They seem to detect the heat just fine, and it seems to elicit those specific behaviors. This summer, I was able to have a student come back to the lab. Unfortunately, COVID kind of did a number on my ability to have undergraduates in the lab, but this summer, luckily, they were able to come back. And I had one student who has really pioneered doing electric shock in larvae. And so she's been giving them 10 seconds of a 20-volt electric shock. And we found that in control larvae, that elicits a very nice sort of C behavior. So the larvae kind of curve up in a nice C. And so now we're testing whether or not NINA-A mutants can show a similar behavior. And so in addition, we asked, okay, are there other behavioral readouts that may tell us whether or not they're responding to a painful stimulus? Because just the initiation of an escape response doesn't necessarily mean that it is being perceived as painful or even something to avoid. And so there are a couple of other ways that we can characterize this. So we know that when they are forced into experiencing a noxious stimuli, they increase their velocity. So they walk around more in order to try to escape. We see this increased stress response as measured by thigmotaxis. So this is that they're spending more time in that outer part of the ring. Okay, here's the increased velocity. And then the other thing we looked at is their social behavior. And interestingly, flies are fairly social. They like to investigate each other. They tend to clump, and actually males do it more than females. And what we find is that we get a decrease in their social interaction as well when they are perceiving this. So we believe that they are perceiving this as a stimulus to avoid, but they are just failing to actually do it. So what I had been hoping to actually show, but didn't quite get to all the final analysis this summer, is that we are now going and looking at single flies, zoomed in into these small arenas where we're basically looking at their electric shock perception. So we're starting at voltages at 0, and then we go to 10, 20, and then we are stepping by 2 volts until we get to 90 volts. And what I should tell you is at about 90 volts, pretty much most of the flies are dead. But prior to that, we have this sort of nice window where we can get some fine mapping of what these animals are doing. And we can quantify actual differences in their ability to detect shock. So at what voltages do they start avoiding it, right? And then when do we start seeing some of these behaviors that we know that we're starting to sort of figure out electric shock initiates? And so some of these are the onset of flipping onto their back. So at a certain point, the voltage gets high enough that flies it's like enough, I don't know if it's a muscle retraction or what, but they flip onto their back and then they'll come back on. They start having these pausing and immobility periods of time. They start showing seizure-like behavior. This is much more prominent in the heat assays that we're doing. We know that certain levels of heat induce a seizure-like behavior. And one of the more prominent ones is this jumps of more than 1 centimeter, where the fly will walk on and then immediately like hop very quickly to the other side of the arena. So coming back to this idea of what is electric shock, if we think that the animal is perceiving it, then we are sort of left with this idea that it's an integration unless there's a deficit in the actual sort of perception level, okay? So how is electric shock actually processed in the fly? So if we go back to that doll neuron that we initially took, and that started us sort of down this road and asked, does this doll neuron in the fly brain play a role in electric shock detection, integration of noxious stimuli? So we did a number of assays where we expressed, in this case, so HID is a transgene. We expressed it in the doll neuron. What HID does is it kills the neuron. And so when there's no shock turned on, they spend as much time as these other flies in the shock zone. When you turn on the shock, even when they don't have the doll neuron, they avoid electric shock. And so that's what you see moving down here. These are the males, these are the females. If you look at the first line, you kill the neurons, they still avoid the electric shock, okay? These are all of our controls. Then we said, well, okay, what if we just block the recycling of vesicles? So we use a Shabiri transgene and we block the recycling of vesicles in the doll neuron and ask, do they avoid shock? Yes, they still do. What if we excited it because maybe it plays some role in excitation? So we used a trip channel that when heated up, this is a sodium channel, it will excite the neuron. Do we see a deficit in the avoidance of shock? And again, we see no deficit. So we feel pretty confident that that doll neuron does not appear to play a role in this actual shock avoidance behavior. So now we've gone on and we've asked, okay, so how else can we start figuring out which neurons are important for this? And so my student, we actually have just received an MRI grant for a confocal over the last year. And so we now have the confocal set up. And so these are larvae actually that express a transgene known as Campari, which is a calcium sensor. And when you shine UV light onto the fly, it will photoconvert that green to red so that you can then image it later on. And so this is in larvae. This is the sort of more central brain area of the larvae, okay? And green is what neurons are active during this actual imaging session. These are living larvae that have been immobilized in agarose gel. And the red was what was active during the shock. And what we find is that we get neurons that light up in response to that shock. What we find is a lot of these neurons appear to be motor neurons. So we get a lot of activation of the neuromuscular junction. And this shouldn't be surprising because we saw in the larvae, they make that nice C behavior. And so there must be some muscle contraction happening. But what I really want to stress is that there's a lot of variability in this. So some of our larvae, we get really beautiful photoconversion and it's really bright and everywhere. And other times we don't get it as strong. We have now, this is in the peripheral area. So this is where the sensory neurons are in the larvae along the body wall. And again, we see some combination. So what you're looking at here are any places where there's yellow or where there's just red would be sensory neurons that were active during this behavior. So since we express this in all neurons, it's actually not super useful. And so one of the things that we hope to do is to sort of start minimizing the populations that we're looking at to try to narrow down exactly which populations. So in summary, what I've told you is that we have these NENA-A mutants that seem to fail to avoid electric shock. At first pass, these appear to detect electric shock, but fail to avoid it, okay? We feel fairly confident that the Dahl neuron is not involved in this shock avoidance phenotype. And while we were trying to understand why these NENA-A mutants failed to avoid shock, we realized that we actually needed to better characterize pain perception in general in flies to really make some conclusions about what is happening with these NENA-A mutants. So for future directions, we're doing many of these assays as well in response to noxious heat, especially in the adult. We have a nice setup where we have half heat and half no heat, and we can also do all of these and it's nicely regulated. And so we can compare the responses in the flies to noxious heat to what we see with electric shock. We're also planning on running painless and some of these other known pain mutants through each of these assays. Our hope is to create a database of responses for many of the common background strains so that we know sort of what we're looking at. And I know there's ongoing work in the mammalian field to do the same thing for common mouse strains. We're also going to break down those neuron populations in order to try to better map that electric shock response in both larvae and adult. And some of these are acquiring lines that we know are involved in noxious stimuli perception. So some of these DA sensory neurons and also the PPK GAL4. And then we're going to also do more work in the adult in a living prep with an open head cavity under the confocal in order to watch live Campari signaling. So with that, I want to thank both current and past lab members of my lab. I have a fully sort of undergraduate lab we had a little bit of hiatus during COVID, but it gave us a good time to also play with some of these now available deep learning algorithms such as deep lab cut and things like that to play with, which has been great. And I specifically want to call out Leora Siegel and Edith Lopez, who've really done a lot of the work this summer on both the electric shock and the imaging. And with that, I will take any questions.
Okay. Let's see, Arturo, I may have lost your image. I'll start with a question. Please, as before, enter questions in the chat. The one thing that I take away from this talk, which is really interesting, is not only how complex behaviors are in flies, but how kind of pleiotropic the genes are in terms of having multiple functional correlates. You mentioned there was some parallel work in in mammals, in mice. Are there any parallels with particular kinds of genes involved in memory and pain in particular?
So you mean like CREB, like the known fly mutant memory mutants? Well, I would say yes, but yeah.
Yeah, I mean, so would you say that this is a particularly fruitful pathway to be going down? In other words, looking at memory-related variants, as well as those things, and see what their involvement might be in various aspects of pain processing, broadly defined.
Yeah, well, so I can already tell you, so rutabaga, which is a mutant in flies that has a fairly, major memory deficit, as well as some of the CREB mutants, are also all known to show some changes in their pain perception. This is what they're finding in mammals as well. When you start looking at these sort of deep, using these neural network programs to look at like animal pose estimation. So when you do really fine mapping of how rodents respond to electric shock, how they lift their leg and their actual sort of motions, we know that in mammals, some of those memory genes are also seem to be, those animals also seem to have some altered, I wouldn't know if it's necessarily perception or integration or sort of behavioral response to that electric shock that had been overlooked previously. And so that is one of the things that we're particularly interested in as well, is going back and looking at some of these memory mutants that have been around for many years. And now that we have a much finer tool to be able to say, do they actually have deficits or changes in how they integrate that painful stimuli? Yeah, so that's sort of where we're going with that.
Amanda, do you know where Nina A is expressed in the fly?
Yeah, so we've tried. So, I can only say sort of bulk where we seem to find it based on, how they've sort of broken up the fly to do RNA-seq work. We don't have an antibody for it. And unfortunately, it's a pretty small gene. And so even doing a few other things, there's some issues. I was recently made aware of a new collection of flies that were all made using, I think, CRISPR in order to try to stick to basically make an enhancer line that we might be able to know where it's expressed. And so I was made aware of that. And so that sort of in the works. And I should tell you, I have an R15, and in my R15, we had suggested making an antibody. We had made a few attempts. It's messy. So I think our best bet is to try to get like a GAL4 expression line to try to figure it out. And even that may not be perfect. Yeah.
What about the homolog, the ortholog in the mouse?
Oh, the PP. Yeah. So that one, it appears to be involved in the immune system. As far as I know, it actually doesn't have that very high expression in the brain. And it's just not well characterized outside of the immune system.
Okay, well, I think we're going to move on to our next speaker, and there was some behind the scenes drama here. Dr. Oriel FeldmanHall was not sure she was going to be able to join us due to IT issues, but she has overcome those. And
the
so
question is, have I overcome these now?
Well, we can see you, and I'll bet we can see your slides once you start sharing them.
My computer is now, my new computer that IT just put up for me is now telling me I don't have Zoom sharing preferences on this IT computer. So
oh, terrific. Do you want to email your slides to me, and I'll run them and I'll share them?
Hold on one second. I think, because I'm signed in twice here.
Okay, well, try it on either one.
I'm going to try something and see if this works. This is also incredible because this morning I had dental surgery, and so I also have a numb mouth. This
is persevering in the face of adversity, and navigating our uncertain social worlds at the same time.
It's true. Okay, so hold on. I'm going to try to do this from my other computer. Bear with me for just a moment, okay?
Okay, sure. And while we're waiting, I'll point out that Oriel is part of the COBRI on central nervous system function, which cannot be accused of having too narrow a focus. That's based at Brown University. And we'll see how this goes. All right.
Okay,
see you again.
Oh God. Can you hear me without?
Yeah. Yep. We can hear you. We can see you.
Ben, are you talking?
No.
Oh, okay. All right. Let's see. All right. And you guys are not having.
There you go. We can see your slides.
You can see my slides.
Success.
You don't have two people. You don't have me twice.
Well, not now. Just well, actually, we do, but it doesn't matter. Just
that doesn't matter as long as you can't hear me twice.
Right.
All right. Forgive me for all of these hurdles this morning, and also I should say forgive me because I'm about to break the mold on all the other talks that have been happening for the last hour. Because I do something completely different. And so I will just do a little legwork in the beginning to orient you to what I do. And then I'm going to stay pretty high level, just given this audience and the fact that I do something so very different. But I'll do my best to integrate some of the work that we do in my lab with what has been said thus far. So here goes. And of course, thank you for having me. Okay, so if we think about our social worlds, our social behaviors occur at basically every level of our social ecosystem. So we have global events like climate change that require widespread cooperation. We also have our daily interpersonal interactions, like who do we trust? And all of these, pervade our everyday lives. This is a hugely complex space. And the fact that social behaviors come in all shapes and sizes makes it really hard to study in static laboratory environments. So we in our lab try to solve this in two different ways. First, to get a better handle on all the different types of social elements and tensions that get baked into these situations, rather than just use one class of behavior, we leverage various different types of behaviors. So for example, decisions to cooperate, trust, punish, or help others. Second, we try to bring as much precision to these nebulous environments as possible while not losing the real tensions that are endemic to the social situations that we might find ourselves. So the way that we do this is we borrow methodologies from a host of different fields. One of those is behavioral economics. And economic games, which I'll talk about a little bit today, allow us to mathematically quantify what people value and how much they value it at. And so we can structure games to mimic real tensions that basically are unfolding in our daily lives. We also rely on theory from social and cognitive psychology. And that we do this is because it informs how to best think about the problems of the mind. So what do we already know about the way the mind is structured that can help us uncover the motivations or boundary conditions for how people behave in social situations? And finally, we turn to neuroscience to help us better understand the mechanisms, some of which we can't always see by exploring behavior alone, that help guide these processes. Okay, so when we consider the ingredients of social behavior, one thing that all social choices have in common is they not only affect ourselves, but they affect other people. So this is a unique property of the social world, which means that we are navigating through great uncertainty at all times. We don't really know what buzzes behind the foreheads of each person that we encounter. What are their intentions? What are their motivations? And because of this, interacting with other people is one of the most inherently uncertain acts that humans embark upon. So think about it, like if we get into a taxi, we're effectively getting into a stranger's car. If you tell a colleague a secret, you're figuring out whether they can keep that secret, whether they'll repay you in kind by not telling the other colleagues on your corridor. You also have to assess what your next action might be in a dangerous situation, or perhaps you're deciding whether to engage in risky behavior with your peers. All of these are examples of situations, social situations that are rife with uncertainty. Okay, so even amongst all of this uncertainty, we still need to navigate our social worlds, and we need to do it effectively. And to do this, we need to learn how to decide, to judge, and learn from the environment around us. So these are the three different realms that we work on in my lab. And I'm going to give you just a tiny glimpse today into two studies that encapsulate the first two, deciding and judging, and how we hope to answer these types of questions. So the first work that I'm going to talk about today is how emotion provides useful signals to bias decisions in uncertain environments. And this work is led by one of my graduate students, Joey Hefner. So all the credit has to go to him for the creativity and the execution for this work. Okay. So there is a basic idea in decision science and neuroscience more generally that argues that value governs choice. So this is a very basic fundamental premise that comes from a larger framework describing how humans and even animals use value to learn about the world to get adaptive behavior. So there's some groundbreaking early work, that of Schultz and colleagues, that I'm sure many of you here are familiar with, where they found that dopamine neurons encode reward. So when there's this expectation of reward that fails to occur, neuronal activity drops, which you can see here in this last image. And this violation of expected reward is called a prediction error. Now, there are hundreds of experiments to date that have documented this process across a wide range of contexts. And it has been very successful in laying the building blocks for how we conceptualize human learning and decision-making. There's a similar framework has effectively been applied to the social domain, where people make these consequential social decisions, such as, let's say, assessing the generosity of another person based on violations of expected rewards. But there's a whole nother field in the past couple of decades that is devoted to understanding the role that emotion plays in decision-making. So this is a very potent motivator of social behavior. So the idea is critical. The critical idea is that emotion contributes to the assessment of subjective value. So if it does that, if we can change emotion, we can change how we value something. And for this very reason, there's been decades of work exploring how emotions such as mood inductions and physiological measurements, heart rate, SCR, self-report, all these different types of methods contribute to changing choice and to changing how we value something. Because there's overwhelmingly good evidence that there's this intimate link between the emotional experience and choice. But there's relatively little, if any, work that has explored the role of emotion and emotional expectations in governing decisions and making choices. So let me illustrate this idea about emotional expectations or violations of emotional expectations with an example. So let's say you're in a department with a lot of other individuals and you avoid collaborating with a certain person because you dread the interactions of this person that you assume is very aloof and grumpy. But you actually have to collaborate with them for whatever reason, and maybe you're on a COBRA, who knows. And what you discover through this collaboration is that the person who you thought was grumpy and aloof is actually quite warm and humorous. And the unexpected joy of working with this colleague therefore produces this positive emotion prediction error, motivating future extensive collaborations with this person. So the idea here is basically that you had this idea that this person wasn't going to be a joy to work with. They actually are. And so there was a difference between what you expected and what ultimately happened within the realm of emotion. So not unlike some other things that we study in the lab, emotion is also a very hard thing to characterize in a laboratory environment or out in the real world. It's been a challenge for decades. And so to help deal with this, we borrowed some of the methodologies used in reinforcement learning, primarily this idea of operationalizing a reward prediction error, where you have the difference between what you think you're going to expect and what you ultimately expect. And we apply that to the domain of emotion. So there's lots of different ways to think about emotion. We co-opted a model of emotion that partitions emotional experiences into two core affective dimensions, which is valence, which we have here along the X, and arousal, which is along the Y. And arousal means like alertness or activation. In our experiments, we use this 500 pixelated fine-grained 500 by 500 pixelated grid where people can move their mouse around to determine how they're feeling, both as a prediction, like I'm going to feel this way, and then in real time as they experience the emotion, the self. Okay, so we embed this measurement into an economic game. So let me tell you about just one game that we've used. It's called the ultimatum game. In the ultimatum game, there are two players, player A and player B. We give player A, let's say $10, could be $100, could be $1, and they have the choice to split their money however they'd like with player B. So in this case, let's say we give player A $10, they offer player B $8, they keep $2 for themselves. Now player B has two options. They can accept the money as is, which means both players take home the money as player A divided it, or player B can reject the money. If they reject the money, then neither player gets any money. So this is a form of costly punishment. It will cost B to make this choice. They won't end up with any money, even if it's small. But what they get to do is they get to punish player A for behaving unfairly. So what we've done in our task is we embed our 500 by 500 pixelated grid into an economic game like the ultimatum game. So for example, a subject might be asked, they'll see a picture of who they're going to be engaging with. This is Thomas, and they'll say, okay, how much money do you think that Thomas is going to give you? And then we have them use this effective grid to place the cursor to tell us how they're going to feel about receiving that offer. And so when they place their mouse on this grid, we can extract a coordinate along the X and the Y dimensions. So let's say 150 by 50, so they're feeling slightly positive and slightly aroused. Then they get the offer, the monetary offer from Thomas. It turns out to be a really crappy offer. They only get, let's say, 10 cents. And we ask them again, okay, so how do you feel now that you've received this offer? They move the cursor to wherever they're feeling, and we extract two more coordinates on the X, Y dimensions. And finally, they make their decision whether to accept or reject. And what this task configuration allows us to do is to compute two different types of prediction errors, an arousal prediction error and a valence prediction error on these two dimensions, given the coordinates that people have clicked on to indicate how they're feeling or how they expect to feel. Now, critically, we can juxtapose these two different prediction errors against the classic idea of a reward prediction error, which is how much did you think Thomas was going to give you and how much did Thomas ultimately give you? And we can see which one of these types of prediction errors is going to win out, so to speak, in predicting these types of social choices. Okay. So what I'm graphing here is decisions to reject, that's to punish player A for behaving unfairly, as a function of prediction error. Positive is indicated by a positive prediction error is indicated by positive numbers, negative prediction errors by negative numbers. And I'm going to graph each type of prediction error separately by color. So arousal in red, reward in green, and valence emotion prediction error in blue. So first, what we find is that if we look at the arousal prediction errors, people who have more arousal than expected end up punishing more. We also find a similar effect for valence prediction errors. If you get less than you expect to get, you punish more, which is not particularly surprising. When we compare these types of prediction errors against reward prediction errors, what we find is that, yes, reward prediction errors are still, in fact, predictive of decisions to punish, but they are at a less significant rate compared to valence emotion prediction errors. In other words, valence prediction errors trump reward prediction errors in predicting these types of decisions. Now, this is in one particular experiment, but we've now run this experiment a number of different times, a number of different studies in the lab, online, and we find a consistent behavioral fingerprint of these three types of prediction errors, no matter how we run the task or even what type of task that we run it in. So the takeaway from this is that there's these unique contributions of multiple different types of prediction errors, arousal, valence, and reward. But critically, the valence, that emotion prediction error, is a more powerful motivator of social choices than reward. Okay. So these data suggest a particularly privileged role for the violations of emotional expectations in guiding behavior of social interactions. But they also have important implications for mood disorders such as depression, which are often characterized by impairments in both emotion and reward processing. And so today it remains unclear which one of these, reward or emotion, is the primary contributor to socially maladaptive behaviors such as apathy or anhedonia. So like in the previous experiments, we reran our experiments. This time we did it in a cohort of healthy controls and those with depression. And what we found is that in the healthy controls, the same behavioral fingerprint that I just showed you emerged again, with valence emotion prediction errors being the most potent predictors of decisions to punish. And when you compare this against those who are clinically depressed, what you see is an attenuation of these valence emotion prediction errors. But what we can do is we can directly compare these two groups on each type of prediction error to see which one of the prediction errors, let's say, diverges or is impaired in those with clinical depression. And what we find is that when you look at reward prediction errors across the two groups, we don't see any differences between how healthy and depressed individuals use reward prediction errors. And that actually dovetails with some previous recent work that found that reward prediction errors are intact in depressed populations. However, we saw startling differences in arousal and valence prediction errors. So those with depression did not use arousal prediction errors at all. And the valence prediction errors were significantly attenuated in the depressed population compared to how they were being used in the healthy population. So essentially, depression was associated with these intact reward prediction errors, but impaired emotion prediction errors. Okay, so why might this be? So we have this other task that we had our subjects do. And in this task, we call it the emotion classification task because what people had to do here is they had to do a really simple task. They had to drag different, excuse me, so now my PowerPoint's not working. Sorry, let's see if I can get this to come up. Here it is. What we had our subjects do is just drag these terms like afraid, anger, which are just emotion labels we asked them to think about what they know about anger, their past experiences with anger, their knowledge of anger, and to use this, this affective grid and place this emotion, afraid, anger, annoyed, and so forth, wherever it might be onto the grid. Okay. So if we look at what that data might look like in a healthy population, for example, you might see that angry is very negatively balanced and highly arousing, and happy, not surprisingly, is on the other side. And so this gives us a good look into the structure of emotion, where things fall along this affective grid. And we can see whether this group of individuals, this is a healthy group, how this compares to how individuals with depression structure their own emotional experience. Okay, so what I'm graphing here, again, is the arousal by valence affective grid broken down by healthy controls and those who are clinically depressed. And what you can see is a pretty startling constriction of those with clinical depression, how they use this affective grid. So this is significant, a significant constriction between these two groups. And essentially what it shows us is that the healthy controls are using or experiencing these emotional terms, or how they think about enthusiasm, aroused, angry, so forth. They're using a much broader arena within this affective grid compared to those who are clinically depressed, suggesting that depressed individuals are displaying this widespread emotional blunting, which may be why they are not using these emotion prediction errors to guide their choices. Okay, so in the last 5 to 7 minutes, I'm just going to talk about more naturalistic ways that we can test this relationship with uncertainty and human social judgments. And one particularly good test bed to explore this relationship is under the hood of something called political polarization, which I'm sure many of you are familiar with, where people routinely express views that are very biased or have these incredibly biased judgments. So the aversion to uncertainty goes hand in hand with wanting to hold stable, predictable beliefs about the world. It's a perspective that is strongly associated with extreme political views. And if you look at our political landscape, those on opposite sides of the political spectrum, in many cases, they're viewing the same information. In some cases they're not, but in many cases they are. And yet they come to interpret that information in very different ways. And the mechanism by which this polarization happens is not well understood at all. So based on some theoretical work, we posited that an individual's inability to tolerate uncertainty would actually drive this polarized perception. And this is work that is led by one of my postdocs, Jeroen van Baar. Okay. So how does the brain transform political information into a polarized perception? So first, what we did is we recruited more than 300 people and we assessed their ideological commitment. And from this group of 360 individuals, we selected 44 liberals and conservatives, and we brought them in to be scanned. And these individuals watched 3 different videos inside the scanner. And this is like a much more naturalistic way to get data, basically, because what they did is they watched a BBC Earth video of beavers making dams. They watched a PBS news segment on abortion that was completely neutrally worded. And then they also watched a political debate between Mike Pence and Tim Kaine from 2016, which was filled with inflammatory language on very hot button issues like immigration and policing. Now, our unbiased metric of shared understanding is something called interbrain synchrony. So let me tell you what that is. Basically, it's a very useful tool for examining how the brain processes naturalistic stimuli, like watching a video. Essentially, it looks for a temporal coupling between brains. So as the brain consumes incoming information, basically the glucose or the bold in the brain rises and falls in a particular area depending on how engaged that area is. And what we can do is we can take that temporal trajectory and we can, in a particular region, in let's say subject A, and we can compare that that activity in the same region to another subject, let's say subject B. And we can see if the temporal rising and falling of the bold signal is similar across these two subjects. Now, another useful thing about interbrain synchrony is we can look at differences at the individual level. So we can combine this interbrain synchrony tool with individual differences like political ideology or a person's aversion to uncertainty to see if these traits that a person holds modulates their neural synchrony, whether it enhances or attenuates, whether they sync up with other people's brains. Okay, so what did we find? So first, we looked for ideological driven brain synchrony. So that is, two Republican brains and two liberal brains being synchronized with one another in all three of the videos. And what we found was in the video, the first video, the BBC Earth video, there was no neural synchrony whatsoever between conservatives and liberals or across the way. Nor was there really any in the neutrally worded segment on abortion, which is of course a hot topic issue. It was just all neutrally worded. Where we saw really robust ideological driven brain synchrony was in the third video, where these two political talking heads were going at each other with like incredibly inflammatory language. So what's critical about this work is that the bold signal linking up between two conservatives and two liberals was on both sides of the aisle. So it wasn't that we were just seeing two conservatives' brain behaving this way or two liberal brains behaving this way. It was on both sides of the political spectrum. And so what you see here in this last image, down here on the bottom right, is in this particular orbital frontal cortex part of the brain, which sits right here. Basically, you can see that two liberals who are in blue have very in sync trajectories of what's happening in that area, whereas two conservatives also have very in sync. So it's not unique to being on one side of the aisle. Now, to get at a very specific question, which is whether uncertainty aversion modulates this ideology-driven neural synchrony, we tested whether participant pairs with similar political views would have greater neural synchrony if they were also uncertainty intolerant. So it's basically an interaction between ideology and uncertainty aversion. And we're looking for synchrony above and beyond the synchrony I just showed you. So this would be modulated by one's aversion to uncertainty. And what we found is that in three different regions, in the TPJ, which is a region that's involved in mentalizing and thinking about other people, the anterior insula, which also came into the orbital frontal cortex, and in the precuneus, in all three of these regions, we saw that uncertainty aversion basically enhanced or exacerbated neural polarization, such that two individuals who were both intolerant to uncertainty and they shared political views produced significantly more neural synchrony in these particular regions. And again, this was not just on one side of the aisle, this was both conservatives and liberals alike. Now, critically, what we found is that the degree to which two brains synchronized in the scanner changed the degree to which they had a polarized judgment after they came out of the scanner. So when we probed our subjects after they came out of the scanner, we asked them how they felt about all the different videos, and we got an index of how polarized their attitudes were. The greater the synchrony between two individuals in the scanner, the more polarized attitude formation they had once they came out of the scanner. So, okay, so I'm at time, which is good because this is my second to last slide. Basically, what I just showed you, just quickly to sum up, is that uncertainty modulates neural synchrony between two like-minded individuals, and this is a mechanism that drives polarized perception. I also showed you that emotionally valenced prediction errors help us make social decisions in an uncertain world, and that these emotion and reward PEs are independent and seem somewhat dissociable, which we're actually following up with at the neuro level right now. So the work that happens in my lab, I am but one tiny cog in a very large system. And of course, my thank you slide is not showing for some reason, or can you see it? You can see it, yes. Okay, so here are all the people in my lab, and I've highlighted the work that I talked about today, both from Joey and Jeroen. So thank you to all of them for doing this amazing work. And of course to you guys for listening and dealing with all of my IT issues.
Okay, thank you, Oriel. You know what I think we might do, actually your talk raises a lot of fascinating issues, but maybe we will go on to Mascha van Wout Frank's talk right now, and then we'll return in the discussion period to asking you some questions. Unless there's something in the chat, well, you're getting applause and other positive comments, very positively valenced comments.
Okay.
One thing that I'd like to ask you when we get to the discussion section is, this morning we had a lot on the vaccine development for COVID and other things, but COVID in particular, and whether you think there are any parallels or lessons to that kind of polarization here. But let's move on. It's a great pleasure to introduce Dr. Mascha van Wout Frank, who works in our COBRE at Butler Hospital, and she has the most evocative title for her talk, which is "The Internal Sunshine of the Neuromodulated Mind. Can Electrical Stimulation Improve the Inhibition of Fear?" I don't know if any of you saw the movie that this is a takeoff on, but that might be worth looking at sometime. Go ahead, Dr. Frank.
All right, so can you see my slide normally? All right, so hello everyone, and thank you for the introduction, Ben, and for letting me speak here today. So I want to share with you what we are working on in terms of developing new ways to treat post-traumatic stress disorder, and specifically about recent efforts on using electrical brain stimulation to improve the inhibition of fear for post-traumatic stress disorder. I will start off though with a little story. I grew up in the Netherlands, and rain, wind or shine, after school and in the weekends, I would play outside with my neighborhood friends. And besides a lot of families on this street, also lived a man alone with his dog. And on plenty of occasions, we would see this man walk his dog. There was nothing really particular about this. The dog was always kept tightly on a leash, but like I said, nothing really noticeable. Until one day when disaster struck. This dog attacked one of my neighborhood friends, and the scene was so horrific that I still shiver even thinking about it. Now, since that day, I approach dogs with extreme caution, if I approach them at all. And that means that even though my friends, family members, and even colleagues keep gushing over the various canine members of their households, for me, that terrifying experience has changed my entire view of dogs. And all I can see is this. Now, I do try to hide my profound distrust of dogs, and with enough effort on my part, I'm able to tolerate some dogs. In almost all instances, this involves very small dogs, and it never really generalizes. Nonetheless, I thought I was fine. I'll never be a dog person, but I'm not too bothered by this on a daily basis. But now I have run into a problem. The thing is that my kids and husband would really like a dog. And as you may imagine, I really do not. I'm even getting not so subtle notes from my daughter depicting something that will never happen, and that is me happily walking a dog while holding balloons. And as for those of you in the audience who have heard my story before, I can say that we still do not have a dog despite the pressure of pandemic pets. So now, of course, I realize that what they see is a harmless creature. Yet I know what such an animal is capable of. So I actually got a lot more insight into my own experience when I started doing research on PTSD at the Providence VA. So, of course, although my story is nothing compared to the trauma experienced by these veterans, the underlying cognitive and neurobiological processes are not that different. Because the fear associated with these types of traumatic experiences generalizes to everything in our veterans' lives. And with the experience of the 4th of July, we heard news on that. Now, fortunately, there are ways to treat this. Exposure-based treatments are the gold standard treatment for PTSD. And during exposure, patients are confronted often through imagination to reminders of the traumatic event. And this activates the fear structure and allows patients to learn that memories and reminders of the trauma are in fact not dangerous and that the stress does not last forever. This in turn should result in the formation of new memories, thoughts and feelings that signal safety. And these safety memories inhibit the fear. Now, exposure therapy actually works well until it doesn't. In 2015, David Morris, a Marine Corps veteran, wrote an interesting opinion piece in the New York Times describing his experience with exposure as emotional chemo, and he couldn't finish it. He's hardly the only one. Up to 20 to 40% of patients who start exposure treatment drop out. And even those who do complete a full course, 20 to 70% of patients report substantial residual symptoms afterwards. And the reason for this is that the fear memory trace will never fully disappear. This is, of course, usually a good thing. We want to fear what could harm us after all. But the core problem in PTSD is that the recall of the safety memories acquired during exposure is inadequate, resulting in insufficient fear inhibition. Now, the process of fear and safety learning can relatively easily be simulated in a lab. Let's say that you see this lamp on a desk, and very quickly you will learn that when the light turns yellow, nothing happens. But when the light turns blue or red, there's a good chance you will receive an annoying electric shock to your fingers. And this teaches you to fear the blue and red lights. This is classic Pavlovian fear conditioning. So now we show you the same light colors, but in a different room. And regardless of the light turning yellow, blue, or red, no electric shocks are delivered. And all light colors are safe. Although overly simplistic, this parallels exposure treatment in that the repeated exposure to the feared lights in this new context results in formation of a separate safety memory that then inhibits the fear. So if you look in the literature about the neuroimaging studies on these types of fear and safety learning paradigms or fear conditioning, fear extinction, they demonstrate that while the amygdala plays a critical role in fear responding, the ventromedial prefrontal cortex downregulates the amygdala during safety learning and recall of safety memories. Yet in PTSD, this downregulation is insufficient due to ventromedial prefrontal cortex hypoactivity. And over the last couple of years, we have been working on augmenting the ventromedial prefrontal cortex functioning using non-invasive brain stimulation techniques with then the ultimate goal to improve exposure-based therapy for PTSD. I'll tell you a little bit about the type of brain stimulation that we're using and some of the experiments that we have done and where this is, how we're developing this. The type of non-invasive brain stimulation that we use is called transcranial direct current stimulation, or TDCS for short. And TDCS uses electrodes placed on the skull to deliver a constant low level of electric current, similar to a 9-volt battery or so, to alter the likelihood of neuronal excitability. And you can, the neuron nicely projected on the brain here. That means that TDCS does not directly cause neuronal activation. Instead, it works in conjunction with endogenous brain activity by giving the brain a little boost so that neurons that just do not reach threshold to action potential, here depicted in blue, now fire when they otherwise would not. TDCS is current dependent. And this means that generally speaking, brain regions close to the anode electrode, and I hope you can see my mouse, here in red, receive an excitatory boost, while neurons closer to the cathode, here in blue, where current exits the brain, receive an inhibitory bias. And here I'm showing you now where we place the electrodes in order to achieve an excitatory bias in the ventromedial prefrontal cortex, given its importance for safety learning and recall of safety memories. And then we used finite element modeling to validate that this was indeed successful, that we could indeed have electrical field current go into this region. And you can see it's a pretty broad region here, all the way from frontal pole to ventromedial and orbital frontal cortex. And red here suggests that the electrical values are higher, so more current, so it's simplistically said. So now to test whether TDCS targeting the ventromedial prefrontal cortex would boost safety learning, we ran an initial experiment in a sample of healthy individuals. So here's the thing. So here we had two groups. One group who received TDCS during safety learning of one light that was initially associated with a finger shock. And then they received sham stimulation or placebo during safety learning of a second light. And the second group got the exact reverse. First, they received sham or placebo stimulation during safety learning of an initial light, and then they received active TDCS during safety learning of a second light. So here are the results. On the x-axis, you will see the number of trials from 1 to 6. Extinction learning or safety learning in humans goes very quickly. And on the y-axis, you see skin conductance reactivity. And skin conductance is a measure of the amount of sweat on your hands and reflects the emotional arousal. And that means here that lower values suggest less threat reactivity and better safety learning. So during safety learning of the first light, both groups showed a reduction in emotional arousal over time, as predicted, and there was no effect of TDCS. However, during the safety learning of the second light, immediately following the safety learning of the first light, the group that initially received active TDCS, but now received sham stimulation, that is here the dotted line, and I'm color-coded when they got TDCS, so blue is when they got the TDCS, and orange is when the TDCS was off. They display a rapid reduction in skin conductance response that then remained consistent over the later trials. Yet the group who first received sham stimulation but now receives active TDCS had greater skin conductance reactivity, particularly during these later trials. So somewhat unfortunately, these data could suggest two things and are to some degree inconclusive. Perhaps the first thing that it could suggest is that perhaps combining TDCS with safety learning might not immediately boost safety learning, but instead enhances future safety in a biased manner, as if it's priming the brain. The second option is that TDCS during safety learning may actually have a slight detrimental effect. So nonetheless, these data were surprising to us. And so we did a second study, this time in veterans with PTSD. And we again compared the effect of TDCS during safety learning versus sham during safety learning. But we also realized that the real problem in PTSD is not necessarily the safety learning itself. It is the memory or recall of the safety learning later. And if we wanted to improve the memory for safety learning, we may instead want to strengthen the consolidation of safety learning into memory. So what we did here, that in the group who received active TDCS during safety learning, we then applied sham stimulation immediately following. And this can be considered the immediate synaptic consolidation phase. So typically consolidation has an hours-long process where overnight you see this what we think of typically consolidation during sleep, but immediately following learning, there's this synaptic phase where the synapses are changing in response to learning. So we thought that may be a fantastic moment to actually try to use TDCS to augment this process. Now, the group who received sham stimulation during the safety learning, we then went on to receive the active TDCS during the early synaptic consolidation phase. So again, I'm going to show you here the results. First, same type of graph, trial on the x-axis and skin conductance on the y-axis. And what we find here is that there's no effect of TDCS on safety learning. And in fact, you can see like everybody's quite relaxed already. But second, when TDCS was applied during safety learning, we observed a return of fear 24 hours later during the safety memory recall. And I want to stress that this is really 24 hours or so later that people come back. And this is a very common phenomenon, and it was this expected. But what was really exciting was that veterans with PTSD who had received TDCS following safety learning did not show this return of fear 24 hours later. And this suggests that TDCS during immediate consolidation prevented this normal return of fear. That's great, we thought. But then we decided to look at the stimulus that was never associated with the shock, the so-called unconditioned stimulus or CS minus. This CS minus or the unconditioned stimulus should not generate any emotional arousal, skin conductance response. But what we found is that when TDCS, when we had applied TDCS during safety learning 24 hours previously, participants now also show fear responding to this never conditioned stimulus. This was not the case for participants who received TDCS after safety learning. So this suggested perhaps that across those two experiments, that TDCS when applied during safety learning could perhaps be detrimental or result in an overgeneralization of fear. And what we should do instead is apply brain stimulation during consolidation after learning. And testing this systematically in a more rigorous manner is precisely what my COBRA project is all about. I'm going to show you a bit about the the setup of the experiment, because I cannot share any data from the project because I'm still blinded in this double-blinded randomized trial. But what I can say is that fear learning, safety learning, and safety memory recall phases are separated over the 3 days to really allow the consolidation of each of these learning phases to occur. And then what we're testing is having 3 groups where 30 participants get, or patients with PTSD receive sham stimulation during safety learning with active TDCS afterwards, or TDCS during safety learning and sham afterwards, or both sham stimulation. So in the meantime, even though my project is a little bit delayed due to the ongoing COVID pandemic, in the meantime, we are not the only group who reported potential detrimental effects of TDCS on safety learning and memory. Just to highlight this for you, of the 7 studies that have tested the effect of TDCS on safety learning and memory, 6, including 2 of our own, reported that TDCS either may or did result in greater fear responses or fear overgeneralization. And I should say that all these studies use skin conductance as their outcome measure in a similar paradigm as I showed you. Now, if indeed true, this could be problematic. After all, we hope to reduce fear responding, not make it worse. We don't really know, because the flip side is that the discomfort or emotional arousal experienced during exposure therapy is actually important to allow patients to learn to cope and for the generation of safety memories to occur. And thus maybe the initial increase in fear responding may actually be beneficial. Like I said, we don't really know. And aside from only 1 study that tested people with PTSD, none of these studies accounted for the fact that exposure treatment, of course, consists out of multiple treatment sessions, and that the time between sessions allows the brain to consolidate into memory what it learns. So what we are also doing is testing the effect of multiple sessions in a study where we combine TDCS with virtual reality-based exposure in veterans with war-zone-related PTSD from deployment to Iraq and Afghanistan. Unfortunately, that latter country is a hot topic right now. So using virtual reality has several benefits. For one, it allows for an immersive environment that includes visual, auditory, haptic, and olfactory input for exposure. Yet we can still control the duration of the virtual reality experience to match it to the duration of the TDCS. And finally, they can control when virtual reality events happen to allow the collection of skin conductance data to assess changes in emotional arousal and habituation over repeated sessions. So to highlight that last point for you, I'm going to show you here a skin conductance tracing of one individual during the first time they went through our virtual reality exposure experience. So on the top, you can see these little gray arrows that signal every time a virtual reality event occurred throughout the session. And you can see below in red that this individual responds with marked increases in skin conductance reactivity when virtual reality events occurred. So when we now look at the same individual, but now having done this virtual reality experience multiple times, we see a clear reduction in skin conductance responding to the virtual reality events, indicating habituation and the generation of safety memories. So in this study, veterans undergo 6 sessions of either active TDCS plus virtual reality or sham plus virtual reality over the course of about 2 weeks. And I'm going to show you here some preliminary data from 12 veterans. On the y-axis are the number of virtual reality sessions, 1 to 6. And on the y-axis is skin conductance reactivity across all virtual reality events per session. So the data revealed a steeper reduction in skin conductance response to the virtual reality events over the 6 sessions in those who received active TDCS plus virtual reality as compared to sham plus virtual reality, despite the observation that active TDCS initially resulted in greater skin conductance reactivity, similar to what we observed in our initial studies. And perhaps more importantly, even though patients who got active TDCS and sham both reported a reduction in PTSD symptom severity from baseline to end of treatment, here EOT in the graph, and we measured it with the PTSD checklist on the y-axis, only patients who got active stimulation plus virtual reality reported a continued reduction in PTSD symptom severity from the end of treatment to the 1-month follow-up. So these preliminary data were encouraging enough to do a larger clinical trial, which we are actually doing right now. The observation that TDCS may cause initially greater skin conductance or threat reactivity remained puzzling to me. And I wondered whether it had anything to do with how the electrical current is distributed in the brain. So we know that with TDCS, the current would hypothetically reach tissues located in between the electrodes. So I'm going to show you here kind of like a figural representation of where our electrodes approximately are. Yet TDCS only uses very weak electrical currents, about, as I said, the strength of a 9-volt battery. So how can we actually get like further deeper in the brain? Because when we place the TDCS electrodes in what we hope would best target the ventromedial prefrontal cortex, and by the way, I'm showing your our montage here again on the screen on the side, we may perhaps also reach the amygdala, which is a region important for threat responding. So to see if this could remotely be the case, we individually modeled electrical field values or fields in various neural regions based on our montage in 34 veterans with PTSD who have since completed this virtual reality plus TDCS protocol. So we first modeled the ventromedial prefrontal cortex here using Brodmann area 10. Although there's quite some variability in the frequency of various electrical field values, and this is actually just a frequency graph, so how often these various electrical field values occurred in response to a 2 milliamp TDCS intensity on the scalp. What you can see is, and I will get back to that variability a little bit later, but what you can clearly see here is that the average electrical field value is about 0.22 volt per meter. But when we then compare these estimates to the estimated electrical field values in the amygdala, we not only saw a very similar pattern, but similar values as well. In fact, there was no significant difference in the average electrical field magnitude observed in the ventral and medial prefrontal cortex compared to what we saw in the amygdala, what was modeled in the amygdala. So this highlights that our chosen TDCS montage allows current to reach the amygdala as a subcortical structure, which, given its role in fear responding, might explain the initially increased fear responding. Now, I should also point out that even though the current may reach the amygdala as a subcortical structure, it was still specific to the amygdala. In fact, the hippocampus, which is located closely to the amygdala, if you look at the electrical field values there, you can see that it was significantly lower, and you're here about 0.19 compared to what we see in the ventromedial prefrontal cortex. And likewise, the average electrical field magnitude and pattern in the dorsolateral prefrontal cortex, which is typically a very well-used brain region for non-invasive brain stimulation because it's so nicely close to the surface of the brain, was actually also significantly different compared to the ventromedial prefrontal cortex. So that kind of suggests to us that perhaps we could get this nice ventromedial to amygdala neurocircuitry that could explain perhaps also the ultimate beneficial effects of TDCS. Because as I mentioned briefly before, and what you saw in those graphs, is that there's quite some variability in electrical field values on these individual, on an individual level. Which of course is to be expected because no two brains are the same, and individual differences in neuroanatomy, such as the folding of sulci and gyri, but also skull thickness and the amount of cerebrospinal fluid affects the current flow. So our next step will be to test whether individuals with PTSD who show comparable electrical field values along the ventromedial prefrontal cortex to amygdala neurocircuitry in response to our TDCS montage will particularly show this initial increase in threat responding together with a subsequent rapid reduction and improvement in PTSD symptoms compared to participants for whom the current may not reach this broader circuitry beyond the ventromedial prefrontal cortex. But of course, first, we need to complete our data collection, and I just hope I will get the opportunity to present those results to you in the future. And with that, I want to thank you for listening, and thank my COBRI mentors, Drs. Noah Phillip and Rebecca Burwell, who continue to show unwavering support throughout this journey, as well as the COBRI leadership, Dr. Ben Greenberg, whose birthday it is today, and Dr. Linda Carpenter, as well as our administrator, Kristen Fortin-Ashburn, and a wonderful team of past and current research assistants, both at Butler Hospital and the Providence VA, and the funding organizations who make it possible.
Okay. All right, Mascha, thank you very much. You're getting a bunch of claps from the virtual hands. I think you almost ended on time, so that does give us a little time for discussion. Because we didn't have it, let's go back to Dr. FeldmanHall and ask questions. But if there are questions for any speaker throughout any of the talks in this session, please put them in the chat or just speak up. And yes, feel free to message me and say happy birthday if you want to. So if we can go back to you, Oriel, the question that I left you with was about polarization in processing or responses to information with respect perhaps to the COVID vaccine, which we spent a lot of time on in the morning in this virtual symposium. Not that issue. That was just obliquely referred to. What we heard this morning was about the actual scientific development of the vaccines and what might come next. So do you think that your work, having individuals look at the video of the vice presidential debate, might have parallels in how people process information about COVID?
Sure. I mean, I think it definitely generalizes to any incoming information that can be processed in separate ways. We're doing some follow-up work on the polarization stuff that I presented on today, both in how people segment information, incoming information. So basically, you know, we get this constant stream of we're basically bombarded with a constant stream of information, and the brain, to make sense of it, has to segment it into distinct we call like neural states or neural dynamics. And how the brain segments this information helps the brain interpret what's coming in. And so what people have shown before us is that, if you're looking, let's say, at a movie, people who will look at the same movie could segment it in different ways and come out with two different interpretations. Now, one of the things that we've done in follow-up work is we found that people segment, incoming political information in regions of the brain, that like Mascha just talked about, like the amygdala, which are very important for emotion and, help with memories of fear and so forth. We find that some of the ways that people segment information are happening in the amygdala, and this has the idea has theoretical roots in something called affective polarization. That polarization comes from a place of fear or emotion. So when we talk about COVID and taking vaccinations, this is, first of all, I need to say, off the bat, this is not my field. This is not what I study. So I'm just opining here and spitballing. But from my read of, what some people are doing these days, the reason that they're not taking the vaccine or they're hesitant is for fear of not enough data, not knowing, what the vaccine will do to the body in the long term and so forth. And so if we apply some of the work that I told you about earlier of how we, let's say either segment information or have we're syncing up with other brains, that might also contribute these, incoming information, taking the same exact information in, but coming out with a different point of view than somebody else may also be happening when it comes to vaccines, sort of vaccine hesitancy. And so the goal should be, if, we're just using the research that I discussed today, getting like-minded individuals to sort of flip themselves and help flip other people to get people to take the vaccine or more readily take the vaccine.
Okay. I don't want to monopolize asking questions of the last two speakers. So if there are others, please put them in the chat or just speak up. Sticking with you, Oriel, one of the things you mentioned, you didn't show the data, I think, but you mentioned that you've done online work. You've done some studies that have been online. And those could be particularly useful during a pandemic because you can certainly collect data. Would you recommend that approach for people with particular kinds of scientific questions? Is there something you can tell this group about what they should be thinking about when it comes to doing online work? Did you find it productive? Were there things you didn't expect?
Yeah, sure. So, I mean, we basically immediately pivoted back in March 2020 to collecting online. I think within the third week of March, we basically pivoted to online data collection, primarily because we had to, right? So we're a lab that can both collect behavioral data and imaging data. And so if we can collect behavioral data in a way that doesn't require us to be in the lab, we're going to find a way to do that. So we were all hands on deck in those first week of the pandemic. Basically, the lab was working really hard to transfer everything that we did or were doing that was not imaging based to the lab. And we were actually able to collect some COVID data, related data as well, which is another thing. But I think it's a great platform. I think it's also great because it doesn't restrict. I mean, in my case, so let's say unlike Mascha's case where she's at the VA, but in my case, our pool at Brown are undergraduates. So we have a very particular type of demographic that comes into our lab with beliefs that are held usually coming from a certain type of background. And so if you're not somebody who would be attending Brown, are we really capturing, let's say, behavioral differences for those populations? And so one thing that collecting online allows us is to expand the type of people that we can collect, not only across age, but also across beliefs and, who might have different traits than the people who are here on campus.
So are you going to be continuing to do that now that things are at least open for the moment?
Well, what we like to do is to do both. So we like to collect a large sample online, like a couple hundred people for a study, which is much harder to do in the lab. It takes a lot longer. But I think it's always good practice to, either do something in the lab with a lot more control and then see if it generalizes to an online sample, and basically publish on both data sets is what we've sort of been adopting these days.
Okay. All right. If there are, again, if there are questions, please plop them in the chat. I'd like to switch to you, Mascha, if I may. So, you mentioned that there's a possibility that when you use anodal TDCS in the context of an extinction learning and extinction retention paradigm, that you've come to the idea that maybe the acute enhancement of the skin conductance response, which might indicate fear, which we think does indicate fear, might be beneficial in terms of the long-term outcome. Yeah. Now, how does that map on to what the behavior therapists would say in terms of the how distressed individuals are during an exposure therapy course, and how that maps on to how well they do?
Yeah, well, that is an interesting question. I mean, I'm not a licensed person. I'm not a psychiatrist or a licensed clinical psychologist. So what I know about this is what I know from others. So I should add that caveat. But my understanding is that having some emotional arousal during the exposure is very important, because you want to have people to learn that they can deal with their anxieties and then cope with it. And if you're not experiencing the emotional arousal, there's not much to learn. So if people are taking anxiolytics to dampen these feelings, then the exposure treatments may actually be less effective. Now, on the flip side of that is you don't want to have people be so distressed that they cannot actually engage with the material and with the treatment itself. So it's a fine balance. And I think that the skin conductance reactivity that we are seeing here is definitely a reflection of emotional arousal and perhaps and threat reaction, especially in virtual reality scenarios. But it's not to the level that people cannot cope. It's not that they're like yanking the headset off and running away. So I think that boosting perhaps that process, and I don't really know. I think this is what we really need to look into. It could also be other reasons for this. This is what I'm currently thinking. But yeah, from a clinical perspective, I think the emotional arousal is critical for having an ultimate helpful treatment response.
Okay. I thought I saw somebody unmute, but I could be wrong there. Well, I think that, Arturo, are there any other questions you might want to ask the group? Do you want to ask our last two speakers how they think that calcium channels fit into this?
That could be fun. No, I do have more of a basic question, sorry for Mascha. I'm trying to understand a little bit the virtual reality setup that you're using. And I'm curious to know how successfully the virtual reality montage that you have is able to somehow replicate the somatosensory experience that someone in Afghanistan or Iraq might be exposed to.
Yeah.
I understand that.
So as you can imagine, there's quite a large range of variation to how people react to the virtual reality. And of course, it's a head-mounted display. We have them sit on a large platform subwoofer. So they sit on a chair. We put them in the position of the driver of a MRAP, so they see their hands. And then they have a head-mounted display and earphones, and they can look 360. So they see a buddy sitting next to them, behind them 2 people, and in the turret somebody. And the reason why we chose for a driving scenario is because they don't have to do anything. Like, you can also do walking, but then people have to move, and we didn't want people to show avoidance behaviors, and therefore we couldn't standardize our virtual reality experience. So in that sense, we have the driving scenario allows them to continue through for 8 minutes, and on several occasions we start slowly with virtual events such as sounds and visual effects, and we essentially blow up IEDs on the sideline. You see smoke, you see fire, and then we have a scent machine where we also send out like sense of smoked rubber, of burning rubber and smoke and fire, etc. So it's very encompassing in that sense.
That's interesting. Well, the other thing that I was thinking is are they I assume they're in a room, temperature control room.
Yes.
And I assume they are not at 50 degrees or Celsius, or like while they are in the desert. The reason of this is because when I've done a lot of some nociception behavior before, and even a difference of 3 to 5 degrees Fahrenheit can have a huge impact.
Yeah, I know.
At least in that pain behavior.
Absolutely.
They normally feel more.
Yeah, no, I agree with you. This is one of the struggles from implementation to do this properly. I mean, one of the things that we are running, we have, we measure the temperature, try to keep it constant. It's a temperature controlled room. We measure the humidity in the room and try to keep it constant. And we end up logging it all and then using it as variables in our analysis to see if we can control some of it out. I mean, you cannot always control everything out, but we can do our best. And yeah, some of it is the case. Like, some people come in and they're late for an appointment and they're running in, or they're doing the first time, they're much more stressed. So you have all these variables you cannot really control, but that's human subject work for you.
or any animal model.
Yeah, exactly.
Well, that's extremely interesting. So as if you needed more variables to control or manipulate, but that's an interesting one. Yeah. Given the given the environment that a lot of.
That's why I'm constantly in our meeting asking about it.
Yeah. Okay, well, we're almost out of time. I'd like to give a plug for the lightning talk session that's happening tomorrow afternoon. There are actually a couple of them that touch on things that we have spoken about. One at 2 p.m. On sort of psychology and health, that is moderated by Victoria Heimer McGinn at Roger Williams University here in Rhode Island. And then Amanda, do you want to talk about your session at 3 p.m. tomorrow?
Sure. We're also doing neuromodulation and I believe neurocircuitry. I have it right here. It's the other neuroscience one. And I've noticed that for those of you who are interested in pain, we actually have a couple more talks, I think, tomorrow, going through that in that session. So yeah, I think it will be good.
All right. Well, I want to thank all the attendees. And you stuck with us through a long afternoon. And Arturo, any parting words for the participants here?
Thank you so much for sharing your exciting science. It was a very fun learning experience these past 2 hours. Thank you.
All right. Hope to see you through some of the rest of this meeting. Take care, everyone.